Hello. Welcome. I'm going to talk about some of the variables that we're going to use in biology this year. So the first two that I want to talk about are independent versus dependent variables, and you've probably heard of these before. I'm going to refer to the independent variable as IV and the dependent variable as DV. The independent variable is the part of the lab setup that we change or we manipulate. The dependent variable is a part of the lab that responds to the independent variable. So we measure this in the lab. Generally, when we make graphs, we use the independent variable on the x-axis, the dependent variable on the y-axis, and we plot our data or information there. I'm going to do an example here. So here's the first example, and this is a biology example that we may use later. So the question is, how does light affect plant growth in terms of height? So the independent variable is underlined in blue, light. The dependent variable is the plant growth, or the height. That's the dependent variable. That's the one that we're going to measure. So we're going to change the effects of the amount of light on and measure the growth. So here's just a simple data table. If we had one bulb, it would grow to 5 centimeters. If we had two bulbs, it would grow to 8 centimeters. And if it were 3 bulbs, it'd be 11 centimeters. So this would be like 1 light bulb, 2 light bulbs, 3 light bulbs, increasing amounts of light. So you can see that it increases the height of the plant as well. So when we graph this, here in blue, the independent variable goes on the x-axis. The number of bulbs is labeled. The light is in parentheses. There's 1, 2, and 3 there for the 1 bulb, 2 bulb, 3 bulb in our data set. The plant height is on the y-axis, 5, 8, and 11. And that's in centimeters. So what I've done is I've plotted in red here where those points are and drawn a line through. This is an example of something that we'll do commonly in the lab. Now, taking and building on independent and dependent variables is we have a control and we have controlled groups. Controls are factors that do not change and are not independent variables or dependent variables. So these things are things that we want to keep consistent or constants in the experiment. The controlled group is a group of, and we'll use an example here in a minute, but these have the independent variable basically taken away. The reason why we have a controlled group is to see exactly if the independent variable has a positive effect or negative effect on what we're trying to measure, the dependent variable. So let's take a look and apply this to our example. So our example is, how does light affect plant growth in terms of height? So again, the independent variable is here, the dependent variable is the height. So the controls, some things that we would want to control or keep constant in the experiment, is how much water are we giving the plants? Are the plants at the same temperature? Soil acidity. Sometimes plants can't grow in acidic soil or basic soil. The type of the plant. So we wouldn't want to compare pumpkin seeds versus corn, for example, because we all know that corn will probably grow taller eventually. So those are some controls, and you can probably think of some others in this case, but those are just some that I listed. The controlled groups are a group of plants that get 0 light bulbs. So in this case, it's probably fairly obvious that the controlled group wouldn't grow very well because it doesn't have any light. We all know that plants need light to grow. So we can then measure exactly how much better does the plant do when it's given light, the independent variable, because we have this controlled group. It's important to do a controlled group in most every experiment that we do. That leads into the expanded hypothesis. So before I talked, there's 3 different hypotheses that we can actually use. The first one we call a null hypothesis. So with a null hypothesis, basically we're making a prediction that there will be no noticeable change in the experiment. So in the case of the light, for example, that the plants would not change in terms of their height. We have, next thing is what we call an alternative hypothesis 1. So this is where the variable x will cause variable y to increase. And you'll see an example of that on the next slide. Alternative hypothesis 2 is the variable x will cause variable y to decrease. So let's take a look at these in our example. So as our example, does light affect plant growth? Height. So the n with a sub-0, the changes in light will not affect the plant's height. So that's our null hypothesis. The alternative one, this is directly related to our controlled group, by the way. Alternative one is the more light, the taller the plants will grow. So that's a prediction that's saying if we give the plant more light, the height will increase. And our alternative hypothesis 2 is the more light, the shorter the plants will grow. Now that might seem kind of silly, but in different experiments, we want to have all of these hypotheses to check for. Sometimes maybe this alternative hypothesis 2 becomes true, and then we have to look at something else in our scientific inquiry piece. That's it for variables today.